Hi guys, I am Ron Persarmiento Belinan and let's talk about translocation. First, what is translocation? Translocation is the process within the plant's functions to deliver nutrients and other molecules over long distances throughout the organisms. Translocation occurs within the series of cells known as the phloem pathway or phloem transport system, with phloem being the principal food-conducting tissue in vascular plants. Nutrients are translocated in the phloem as solutes in a solution called phloem sap. The predominant nutrients translocated their sugars, amino acids, and minerals, with sugar, sucrose being the most concentrated solute in the phloem sap. Various cell types utilize these nutrients to support their requirement for life or store them for future. Next is the transporting tissues. Water and inorganic substances absorbed by the roots have to be transported to the leaves through xylem. Likewise, sugars and other organic compounds assimilates synthesized from the leaves have to be distributed to all parts of the plants through phloem. First, let's identify phloem and xylem. First, phloem is largely for the transport for assimilate sugars. However, other substances such as growth regulators, nucleotides, some inorganic minerals, and systemic pesticides are likewise transported via these tissues. Next is xylem is essentially for the transport of water and minerals. Direction of movements. Sugars, which are synthesized during photosynthesis, move throughout the plant, principally through the phloem tissues. The movement can be downward or called basipetal from leaves to roots, but lateral and upward movement acropetal from leaves to buds, or to fruits to storage organs also occurs. Direction of flow of water in xylem is commonly acropetal in both tissues, xylem and phloem. Lateral movement of substances is possible due to the presence of plasmodesmata. Mechanism of translocation. Photosynthetic products are moved out of the leaves and into the assimilate stream from the source. An exporter organ or tissue that produces assimilates to when they are used or stored called the sink, importer or consumer of assimilates, as you have seen in the diagram.
Phloem's transport is believed to occur by mechanisms of pressure flow. This hypothesis suggests that assimilate are moved from translocation sources to sink along a gradient of hydrostatic pressure of osmotic origin. Sugar is asserted to be transported in the phloem from adjacent cells in the leaf by an energy-dependent active process called phloem loading. The effect of this process is a decrease in water potential in the phloem sieve tube under osmotic pressure. The water then acts as a vehicle for the passive transport of the sugars to sink where they unloaded or removed for use or storage. The water is circulated in the transpiration stream because of the increase of water potential or the sink resulting from the phloem surrounding. Assimilation is the process of utilizing food, photoassimilates, and other solutes for growth. During the early stage, food substances are converted into simpler compounds and used as building blocks for more complex substances. During the later stage, simple and complex compounds are integrated into the living substances of the cells. Movement of assimilates. Upper leaves supply the assimilates for the stem apex while the lower leaves supply the roots. The removal of apical leaves induces the lower leaves to send assimilates to the leaves above them. Always remember that active sink is fed by the nearest source. Factors affecting partitioning of assimilates. Number 1. Sink strength. This is the ability of a sink to accumulate assimilates. It is a function of sink size and sink activity. Number 2. Proximity of the sink to the source organ. It assimilates move preferentially toward sink above and in the line with the source. Lower mature leaves feed mainly at the roots, while the higher mature leaves feed mainly at the young leaves and the shoot apex. Number 3 is the stage of development. It is the developing flowers and fruits become dominant sinks during the reproductive stage of crop. On the other hand, storage roots used as planting materials export assimilates to developing vegetative tissues. And lastly, nature of vascular connections between source and sinks. Each leaf is connected to the main vascular system of the stem by a vascular trace, which diverges from the vascular tissue of the stem into the petiole. Translocation is the long-distance transport of food materials within a plant. As shown in the figure, glucose produced by photosynthesis in leaf is converted to sugars and translocated to different parts of the plant, to growing regions to be used as energy for growth, to storage organ to be stored mainly as sugars, and lastly, to storage organ to be stored mainly as starch. Also, translocation is the movement of organic solutes, example sucrose, from a source to a sink through the phloem by means of mass flow source, wherein source and exporting organ. Number 1, storage organ during export phase. Number 2, matured leaves. Sink In sink, any non-photosynthesis organs that does not produce enough photosynthetic products to support their growth and development, like apical regions, immature fruits, and developing storage organs. In companion cells, each sieve element has at least one companion cell alongside it. Companion cells have a similar structure of a normal plant cells, as they have a cellulose cell wall, a plasma membrane, cytoplasm, a vacuole, and a nucleus, but they have a larger number of mitochondria and ribosomes, as these cells are very metabolically active. Also, the vacuole of a companion cell is relatively small instead of large. Many plasmadesmata pass through the walls between sieve elements and companion cells, providing a direct pathway between the cytoplasm of the companion cell and the cytoplasm of the sieve element. Sugar loading and unloading. How is sugar actively pumped into the phloem? There are two known mechanisms operating in different species. In one, sucrose enters the cell walls near the phloem in the smallest veins of the leaf. It then enters the phloem by attaching to sucrose transporter proteins embedded in the plasma membranes of the sieve elements and companion cells. In the second mechanism, sucrose enters the companion cells of the minor vein through small plasmodesmata, and it is converted to larger sugars, raffinose and stactose. These larger sugars are unable to diffuse back through these plasmodesmata due to their size. Therefore, they are trapped in the phloem of the leaf and build up to higher concentration. They enter the sieve elements through larger plasmodesmata and are carried away toward the sinks. When sugars and other nutrients arrive in the sink tissues, they are unloaded from the phloem and enter surrounding cells, either throughout plasmodesmata or by crossing from one cell to another across the cell walls. The size and metabolic activity of the different sinks determines the amount of material that is delivered to them. Thus, the use of sugar in the sinks determines how much sugar flows to them. So, there are two transport systems in plants, the xylem and phloem. Xylem is transport water and minerals salts no need for energy as ATP made of dead cells, and the phloem is transport food. Example, sucrose amino acids using ATP made of living cells.